I'm Emily Grisbeck. I am the President-elect and Programming Chair for Minnesota IFT. So thank you all for attending today, our Macy Award and Student Recognition virtual meeting. Have a couple of quick housekeeping things. First is that we are recording this presentation, so know that, and that we will be posting it for access later, and more information will be coming out after the meeting today. Please do mute yourself during the presentation. We will have a Q&A session for our speaker today, so feel free to unmute yourself during the Q&A, or at any time utilize the chat box to drop a comment, and we can read those off when we get to the Q&A portion as well. And with that, I'm going to turn it over to Hasmukh to present the Macy Award today. Hasmukh, you're muted.
You have to unmute yourself.
Oh, thank you, Emily. And thank you, everyone, for joining us today for this important event, Macy Award presentation. Normally, you know, it's so much excitement when we get together in a room about 150, 200 people. It's a lot of excitement. But because of the COVID, I think we are not able to get together. But this is a virtual event. And so I would just like to, my name is Hasmukh Patel, and I'm the chair of the Macy Award Committee for 2021. So I just want to give you I think most of the people know what the Macy Award is, but just a very brief introduction for the benefit of for the benefit of the people who don't know much about the Macy Award. But Macy Award was established, you know, it's a Harold Macy Food Science and Technology Award, as full name, was established in 1981 by Minnesota section of IFT. Dean Macy, it's in the name of the Dean Harold Macy, and he was the Dean of the Institute of Agriculture, Forestry and Home Economics Department, and was the founder member of the IFT. And here is, you can see the picture of Dean Macy being honored as an inaugural recipient of the Macy Award. So just a little bit, you know, in recognition of the Macy Award is actually presented in recognition of the outstanding, as an outstanding example of food technology transfer or cooperation between scientists or technologists in the field of academia, government, or private industry. So, you know, normally we would present this award as a, you know, to Dr. Schaffner today as a, you know, as a plaque. But this is what the plaque look like today. You can see, and this award is presented to Dr. Schaffner, Dr. Don Schaffner today, in recognition of his outstanding research to facilitate the adoption of predictive food microbiology and risk-based thinking in government and private industry to support the science-based risk assessment and assurance of the food safety. Next. And I would like to recognize the Macy Award Committee 2021 this year. George Anor, he is also a president of Minnesota section of IFT this year. Emily Grisbeck is president-elect, and you just heard her. Dr. Kumar Mallikarjunan, Anup Kolanor, and Brenda Polzin. So thank you, everyone, on this committee. We had a very good committee and a lot of hard work by the committee. Thank you. So this year, as I mentioned, that this award is being presented to, we are so, it's pleasure on behalf of the Minnesota section of IFT to introduce Dr. Donald W. Schaffner. He's a distinguished professor and extension specialist at Department of Food Science, Rutgers University, New Jersey. And just by the way of a little introduction, he has a, you know, there's so much things can be, you know, we can say about Dr. Schaffner, but just to give you some highlights, Dr. Schaffner's research interest includes quantitative quantitative microbial risk assessment and predictive microbiology, food microbiology. He has it's close to 200 peer-reviewed publications, several book chapters, and he also got at close to $10 million in competitive grants. And Dr. Schaffner, by the way of his training and education, he has trained and educated thousands of food industry professionals through his short courses. In fact, he has some actually podcast and on the microbial food safety. And if you look at the brochure of the program, the links are already mentioned, and I believe that he will also mention these podcast links in his presentation as well. So in addition, so far Dr. Schaffner has been also awarded with several other prestigious awards, including IFP awards, and he's also been serving as an expert on several international committees and national committees, including U.S. National Academy of Sciences, World Health Organizations, Food and Agriculture Organizations of United Nations, IFT, and U.S. National Advisory Committee on Microbial Criteria for Foods. So much can be said about Dr. Schaffner, but I would not take too much time today, and you can read this in a brochure. With that, introduction, I would hand over this to Dr. Schaffner for our presentation.
Thank you so much, Hasmukh. I appreciate it. I'm going to go ahead and try to share my screen, and then I'm going to put this into presentation mode. Let's move some things out of the way. So thank you so much for that introduction. I'm in an effort to make some connections. The Weber Laboratorian Award unofficially comes with a vintage microscope. You can see that microscope over my shoulder there. I need to work on finding the spotlight or something. It's sitting in the dark there. And I also want to say apologies to those of you who haven't seen me in a while. I'm sporting my COVID haircut. You can see a little bit of my COVID haircut in the promo picture, which was taken during the summer, but I have not had a haircut since the pandemic started. But I am due for my second dose of the Pfizer vaccine next week. And so 2 weeks from then, I'm going to consider getting a haircut. So the title of my talk is Application of Predictive Models to Manage Microbial Risk, but I've given myself the subtitle of the Minnesota edition. So first of all, it's my extreme honor to be selected to win this award. And it got me thinking, wouldn't it be cool if I could give a talk where I mostly talked about stuff that had some sort of connection to Minnesota? And so that is this talk. This is a variation on a number of talks I've given over the years, but this is the only Minnesota, fully Minnesota edition of this talk that I've ever given. So let's get into it. So I wanted to provide a bit of a historical perspective, and thanks to my mom for this. She just texted me out of the blue this picture that you see over here on the right. That is a very young looking Don Schaffner without a COVID haircut. This was an article that appeared in Food Technology magazine back when we used to read things that were on paper. And what I want to point out to you, and this is from Education News, I think it's from 1989. I want to point out my research interests, okay? When I joined the faculty at Rutgers, I naively put my research interests as traditional and non-traditional food fermentations. That's what I did for my master's. Utilization and recovery of food processing waste. That's what I did for my PhD. Food safety and sanitation, because as a food microbiologist, what else am I going to do? And then microcomputer applications in food science. You don't see any mention of hand washing or risk assessment or predictive modeling, but I think you can maybe see in that phrase, microcomputer applications in food science, perhaps the beginning of a germ of an idea that turned into the rest of my career. I don't think if you'd asked this guy in 1989 where he would be in 30 plus years hence if he would have predicted that I would be sitting here talking to you guys today. So anyway, let's move on to the next slide. Why am I a predictive food microbiologist? Well, I think if you had to blame one person, this is the first Minnesota connection, if you're keeping track. If you had to blame one person, it would be Ted Labuza. I still remember to this day sitting in a classroom in Rutgers, hearing Ted come and talk about the work that he did on TTIs. And the idea, it still gives me goosebumps to think about right now. I had an idea. I was like, wow, like this is what I've been looking for, right? Ted talked about microbial food spoilage. You talked about TTIs, and I said, you know, this is what I've been looking for. This could be something that I could do at Rutgers. I like math, I like statistics, I like microcomputer applications in food science, apparently, and this could be something I could do. And so I credit Ted for turning me into a predictive food microbiologist and being able to use the my interest in microbiology and my aptitude for statistics and math to launch my career. So I'm forever grateful for Ted. And then I began to do predictive food microbiology. So that was, let's say, flash forward now about 10 years. This is now the second Minnesota connection. So I was asked to be part of a team that was funded by the IFT. So nice connection there with the IFT section of Minnesota, of IFT section in Minnesota there. I was tasked to be a part of this group to work on the evaluation and definition of potentially hazardous foods. And to show you, again, the way this has worked its way into my life, I was on a conference call today talking about TCS foods with AFTO, the Association of Food and Drug Officials. And so potentially hazardous foods became TCS foods, more about that in a minute. But this continues to be a big and interesting part of my life. So we deliberated through this time frame, which includes the events of 9/11. I've got funny stories about 9/11, not funny stories. I've got stories about 9/11 that relate to this committee that I won't talk about. But we issued our report, our final report, on December 31st, 2001. So where's the Minnesota connection? You might be asking yourself. Well, this one has two Minnesota connections. The panel was chaired by Frank Bushta, who I think is here on the call today. Hi, Frank. Good to see you virtually. And Frank, I think, picked me to be part of this group. And again, I'll forever be grateful for the profound effect that's had on my career. And then again, a giant shout out to my good friend and colleague, Katie Swanson. This was the first time I met Katie, and we've since worked together on a number of occasions. And it's always great when I can get a chance to work with Katie on something. So this group was charged with a variety of things, including, to answer this question, are processing technologies or mathematical models sufficient to deal with whether something should be a TCS food? And so I think this is part of the reason why Frank wanted me to be part of this group. So this has been 10 years since I started at Rutgers. I was making a bit of a name for myself in doing predictive modeling. And so I was able to hopefully help this group think about models in the application of this idea of defining what it means to be a TCS food. And I hope you can also see the connection with the Macy Award here, right? So we've got individuals from academia, we've got individuals from the food industry working together, people like Frank Yanis, you may have heard of him. He's eventually ended up in the federal government. Individuals working together under a charge from FDA to develop this guidance document, or this report. And so again, very much, I think, in keeping with the theme of the Macy Award. So here is just I cheated. I did a search for the word model within the entire report. And you can see here is a paragraph from the executive summary. I won't read it to you, but it talks about the application of computer models in this idea of potentially hazardous foods. And so what we were able to do was to create these two charts. Again, giant shout out to Katie Swanson here. I remember sitting somewhere, it might have been a conference room at Walt Disney World. It was certainly somewhere working together to pull data from the scientific literature to construct these two charts. And what these charts or these figures show is the interaction between water activity and pH and growth and no growth conditions for a variety of organisms. On the left-hand side, you can see spore-forming organisms. So these would be organisms that would be present in foods that have been cooked to kill off the vegetative cells. And then over on the right-hand side, you can see the same sort of plot for vegetative organisms of importance in food safety. And so the committee essentially used these data to define or to redefine what was potentially hazardous foods and what was going to become TCS foods. And so again, I had a lot of fun on this committee, made some great connections and some great contacts with folks, and we issued this report. And it was I think a useful report. The report actually eventually went on and modified the food code, which is another example of this interface. So this got me interested in the Conference for Food Protection, which is where academics, government folks from state and local and federal government and industry come together to help the FDA decide what's going to be in the food code. And so the work that we did as part of that PHF group actually became part of the food code. And so you can see, I think, a relationship between those figures that I showed you and these tables. Again, here we're looking at an interaction of pH and water activity, and whether something is a TCS food or a non-TCS, or whether something is a non-TCS food, or whether a food needs a product assessment. So this is table 1 from the food code, which looks at vegetative cells, and here's table 2, which looks at spore-forming organisms. So again, a lot of very, very nice memories, and doing hopefully doing good to improve food safety and improve food safety decision making when it comes to food safety regulations. So what's the next step? Well, the next step for me, in terms of this story, was being appointed to the National Advisory Committee on the Microbiological Criteria for Foods, and working with a bunch of colleagues to develop this document, which was published in the Journal of Food Protection, entitled Parameters for Determining Inoculated Pack and Challenge Study Protocols. And I put this slide in because I always think of the MNIFT document as being the next step forward from the FDA IFT document. But then I said, well, wait a minute, I'm not sure there's a Minnesota connection. But then I got to thinking, I looked up the people that worked on it, and in fact, there is a Minnesota connection here as well. And it was Tim Fryer, who was part of NACMF at that time, who worked with us in writing this document. And so Tim at the time worked for Cargill and was, and I think still is, a resident of Minnesota. So even here, we've got a Minnesota connection. And again, performed the same cheat code. I opened up the NACMF document, which along with the IFT documents are essentially two documents that I have on speed dial on my computer. I refer to them on, if not a weekly basis, at least a monthly basis. I opened the NACMF document, I did a search for the word models, and you can see here, this is what FDA asked us to answer. What are the appropriate uses of mathematical growth and inactivation models? Under what conditions can they be used as a substitute for inoculated pack and challenge studies? And then of the models available, which ones are the most suitable and what are their limitations? So I won't talk too much about that, except to say you can go and read the report. Should be freely available on the internet if you want to learn more about that. And I will say I went on with my colleagues Linda Harris from UC Davis and Kathy Glass from the University of Wisconsin-Madison to develop a short course for teaching people about the NACMF document. If Kathy Glass had only moved one state over, I could claim her as a Minnesota connection, but at least it's Wisconsin, which I think is close. So let's go on to the next slide. So I want to talk about continued policy applications for mathematical models. And so what you see over here on the right-hand side, I mentioned the Conference for Food Protection before. This is a document that was developed by the Conference for Food Protection Committee on Emergency Action Plans for Retail Food Establishments. And so you can see here another interaction grid. Here, it's not pH and water activity, it's maximum product temperature and it's time. And you can see here the relationship between temperature and time looking at whether if it was okay under this emergency action plan to hold, sell, and serve a food, or whether the food needs to be either cooked or discarded or returned to temperature control. So I had to stretch a little bit here to make a Minnesota connection. The deliberations of this committee were spurred in part by Superstorm Sandy. Superstorm Sandy decimated the East Coast. Where was I during Superstorm Sandy? I was in Minnesota, and not having my power go out and not having to deal with all of the terrible things. I shouldn't laugh, all the terrible things that went on during Superstorm Sandy. So I was there for a work-related trip. My wife had the good fortune to be there with me, and we made the decision, hey, why not extend our stay in Minneapolis, St. Paul? Because we sure didn't want to go back to a house in New Jersey that had no power and had spoiled food in the refrigerator. So that's a bit of a stretch, but there's a Minnesota connection there as well. Okay. So, and this has no real Minnesota connection, but I want to talk about the continued policy applications and talk about how the work that I did for all of these committees led to application in my laboratory. So this is some work that was done with a graduate student of mine, Jen McConnell, who's from New Jersey. She's not from Minnesota, sorry, looking at validation of mathematical models for salmonella in raw ground beef under dynamic temperature conditions. And so the abstract is less interesting than the figures. Again, we don't really have time to talk about this in detail, except to say that here are the data that Jen collected together with mathematical models, looking at, could we predict, using existing models, could we predict the growth of salmonella under changing temperature conditions? And it turns out the answer is we can. I continue to use models like this every day in helping food companies manage food safety decision-making. So again, if you're interested in more detail, by all means, check out our paper, which was published in the Journal of Food Protection. Okay, I'm going to change gears here briefly, and I'm going to sort of take you down a different tack on the use of mathematical models to help with making policy and making food safety decisions. So we're going to change gears, and we'll change gears a couple more times throughout the presentation this afternoon. And here I want to talk about now the cooling of cooked foods. And so, as promised, there is a Minnesota connection here as well. And so my interest in Clostridium perfringens, and specifically in the cooling of cooked foods, can be traced to a number of individuals, including Vijay Juneja, who's with USDA. No Minnesota connection here, but I think certainly many of you on the call will recognize Oscar P. Snyder, or as we call him, Pete Snyder, sadly now departed from us here. And Pete, I still remember, again, many conversations with Pete at IFT meetings, talking about all of the wonderful ideas that interested Pete, that had a unique intersection with many of my own ideas, to talk about, in this case, about cooling. And so Pete got me really interested in looking at the safety of cooked foods during cooling. The work that he did here with Vijay and with Miriam inspired me to submit a USDA grant proposal that was funded, which led to our own work with Clostridium perfringens. And to this paper with Sarah Smith Simpson. Sadly, there's no Minnesota connection with Sarah. She's from Michigan, but you know, it's another state that's up there in the same part of the country as Minnesota. So you know, maybe you'll claim Sarah as well. Sarah was a wonderfully productive graduate student in my laboratory. We published a number of different papers, including this one entitled "The Development of a Model to Predict the Growth of Clostridium perfringens in Cooked Beef During Cooling." Again, don't have a lot of time to go into that paper, but I will share with you this particular figure. We looked at different cooling regimes. We looked at computer models. We actually made a computer model in this paper. It turns out the model we made was not nearly as good as one that somebody else made. The perfringens predictor model that's a part of Combase is actually a much better model. Our model ends up being very, very conservative. That is, it ends up being very, very fail-safe. I prefer the perfringens predictor model as being a better model for predicting the growth of Clostridium perfringens. But again, I always will credit Pete for getting me interested in the cooling of cooked foods and the food safety implications of that. And that story is not over yet either. That story continues to this day. You can see here an abstract of a paper that I published with my graduate student, Matt Igo. Matt at the time was a master's student, now a PhD student. He's actually going to be finishing up hopefully this summer. And this is a paper that was not at all part of his PhD dissertation or his master's thesis, but was something that he took on as a side project entitled Validation of a Simple Two-Point Method to Assess Restaurant Compliance with Food Code Cooling Rates. And this is an outgrowth of another paper that I published with colleagues at CDC. And so again, I hope you can see that connection between academic research and then with policy and with regulation, with the collaboration with CDC. That CDC paper inspired me to collaborate with Nicole Hadeen. And again, here we've got yet another Minnesota connection. Note that Nicole is part of the Minnesota Department of Health. She came to us with some data sets that we could use to apply the ideas that we created during our initial collaboration with CDC to see if we could use this method to assess cooling rates in restaurants. So a bit of a digression here. We know that restaurants often have difficulty cooling cooked foods. Inspectors, and the food code says you have to cool over 6 hours with certain time temperature parameters. Inspectors are not going to be inspecting a restaurant for 6 hours. And so the question that we asked was, well, is there a simple way that inspectors could take limited temperature measurements during their visit to a restaurant, use those limited measurements to assess the cooling rate? And the short answer is yes, it works, at least under some conditions. And so, and again, this collaboration continues on to this day. I've been working with folks from CDC again, as well as other colleagues from CDC to further flesh out this idea and to basically see if we can further refine this idea. Can a restaurant inspector go into a restaurant, take a limited number of temperature measurements of cooling cooked foods, maybe once when they arrive in the restaurant and the second time before they leave, and then use those simple two-point measurements along with some growth kinetics and some cooling kinetics that Pete Snyder taught me to basically determine whether a food was going to meet or not meet the food code, the state food code cooling requirements. And so again, more to come on this particular topic in future publications. All right, I am once again going to change gears and talk about a different topic. And this is just a brief mention, probably could say so much more about NCFPD, the National Center for Food Protection and Defense, which I was part of. Which was housed at the University of Minnesota. And then we used in this paper, which was published with a couple of Canadians. Well, Emma's originally from the UK, but lives in Canada now, Emma Hartnett and Greg Powley, to look at, could we model the public health response to a terrorist event in the food supply? Data provided by Craig Hedberg, another Minnesotan, was very helpful to us in doing this work. I have to say, of all of the things that I've done, the thing that probably brought me to Minnesota the most often on a regular basis was being part of the National Center for Food Protection and Defense. And so thanks to Frank and others for making me a part of that and inviting me to be part of NCFPD. Always have very fond memories of all of my times in Minnesota, mostly in nice summer months, to be part of NCFPD. All right, changing gears. This is for the last time, and then we'll take it into the wrap up here, and I'd be happy to have questions and comments. I want to talk a little bit about flour risk assessment. So I was approached by a company who shall remain nameless that you might recognize, as being another Minnesota connection, to begin work on risk assessment of flour for E. coli and Salmonella. And so we did some work on that, which I'm not going to talk about, but I am going to talk a little bit about some interesting stuff that we got that intrigued me as a result of getting interested in flour risk assessment. And so what you see over on the right-hand side is data from CDC. And this shows data on people infected with the outbreak strain by date of onset. And so what you can see is that you see on any given day, you might have 1 or 0 or 2 people getting sick with the outbreak strain from flour. And so it occurred to me that maybe with a little bit of data analysis and manipulation, we could test the hypothesis. And the hypothesis, which is again based on what I know about flour shelf life, essentially the way the flour distribution chain works is the consumption of the flour, disappearance of the flour from the distribution chain is essentially linear over time. And so if you put a package of flour on the shelf and then somebody buys it, it's basically a uniform distribution over time, over approximately 9 months or a year before that flour is used. And so I said to myself, so if that's the case, and if E. coli dies off linearly with time, which at least some of the published data seems to indicate, maybe we can validate, or we can figure out what that die-off rate was in the impacted flour by reanalysis of this data. And so again, this is another example of a collaboration. We've got data from CDC. We've got an academic, myself, doing some risk analysis or risk modeling to perhaps help the flour industry. And it turns out that you can, in fact, do this. And so if you take the CDC data that you see here on the right-hand side of the slide, and you regraph it, and you look at the number of illnesses over time, where you bundle those illnesses into 50-day intervals, what you can see is that at time 0, there's no illnesses. During the first interval, you see a bunch of illnesses, and then essentially those illnesses trail off over time. And so the question is, could you take this data and then figure out whether that data somehow corresponded to, would enable you to predict a die-off rate for the organism? And in fact, with a little bit of mathematical modeling, you could do that. You could figure out that what the CDC data predicted in terms of the number ill versus if you took this linear decline data, predicting the, sorry, just one second, I'm going to close the door to my office. So what you can see here is that there is essentially a correspondence between the CDC data, right, which is simply data on people getting sick, and the estimates of the model, which uses a dose response model for the microorganism of interest, E. coli 157H7, and basically guesses at what the decline rate would be over time. Now, the problem with this analysis is the decline rate that this sort of back-end or re-engineered model comes up with is about an order of magnitude different, slower than the published decline rates for the microorganism. And so what that says to me is, hey, that's a research question, right? What do we know about those papers and how they inoculated the E. coli into the flour? Is this an indication that there was perhaps something about the E. coli in this outbreak flour that actually makes it more resistant to inactivation over time, right? Maybe there were, I don't know. I mean, when we've been fiddling around with this a little bit with Matt Igo, who I mentioned before, with his PhD dissertation, we've been looking at how you culture E. coli, or actually in his case, how we culture Salmonella, and how that influences the resistance of that Salmonella to inactivation in different dry food products. And so I think, again, this is not an answer so much as it is a question and a hypothesis that needs to be tested in the future. So yeah, anyway, more to come on this topic as well. So I usually talk at about 2 minutes a slide, and I can tell that I've been very excited today, and I've talked at a faster rate than 2 minutes per slide, but that's going to leave us hopefully lots of time for questions. In terms of answering the question, what about the future of using predictive models to manage microbial risk? Well, I think as Mr. Spock is indicating here, the future is bright. I'm not sure we're going to all wear shades like Mr. Spock is wearing here. Those look like they were fashionable maybe in the 1960s, but I think certainly the future is bright when it comes to the use of mathematical models and microbial risk assessment to manage risk in the future. As Hasmukh indicated, I will be plugging my 2 podcasts. I do 2 podcasts that I co-host with my good friend and colleague Ben Chapman, and you can find out more at foodsafetytalk.com and riskyornot.co. Food Safety Talk is a rambling podcast that's mostly about food safety that comes out every 2 weeks. Risky or Not is a newer podcast that comes out 3 times a week with much shorter episodes, about 10 or 15 minutes, where Ben and I decide in Siskel and Ebert fashion with thumbs up or thumbs down, whether something, probably something proposed by one of our creative listeners is in fact risky or not. So with that, again, my profound thanks to all of the Minnesotans that I've collaborated with over the years, and to the committee for inviting me to give this Macy lecture here today. I'm just sorry that I can't be with you in person. I dearly have loved all of my visits to Minnesota and to Minneapolis, St. Paul in particular, and I look forward to being able to safely travel again in the future and joining you all in the real world. So thank you. With that, that concludes my presentation. I would be happy to answer any questions.
Thank you, Dr. Schaffner. Very enlightening, very impactful. I think there's so much impact your work has brought to the world. I think it's yeah. We will just take any question from the floor. It'll be nice if anybody has a question directly can ask to Dr. Schaffner, or otherwise we'll have a question from a chat. You can also put the question in the chat and we'll pick it up from there too. Any question for Dr. Schaffner? Okay, I'll start with something, Dr. Schaffner, and then maybe meantime this question. So in terms of, I think you have done so much work on the cooling, the impact of cooling and all those things. What is there anything, can you give us a kind of a glimpse on what is coming next, like what you're working right now and what, the predictive models can be applied to anything in future? What's your, if you can give us some highlights.
Sure. I'm still really interested in this validation of the 2-point method, right? And so the data that Nicole Hadeen provided from some work that she did there in Minnesota was good, but I'm looking for a larger data set. And then as a result of re-engaging with Nicole and with Laura Brown-Green from CDC to kind of get out the word about cooling and the 2-point method. I met a colleague from CDC who actually was able to provide me with a remarkable data set with 99 different cooling curves based on cooling data he collected while he was basically working to assure public health in the nation's national parks. And so many people may not know, but if you're a national park, you're not inspected by the state, you're inspected by the feds. And in fact, it's the CDC that does those inspections. And so this was a guy who was really interested in cooling, and he collected these cooling curves over a long period of time. And so he said, hey, I've got all this data. Could you do anything with it? I'm like, absolutely, I can do something with it. And so we've got this very large data set of 99 cooling curves. And so we're looking at different ways to analyze those data to both determine whether they complied with the FDA model food code cooling requirements or not, but also, in addition to that, taking those data and plugging them into Perfringens predictor, which is the model that I use for predicting Clostridium perfringens growth during cooling. And then we also have some models that were developed by my colleague Mishra, who is currently at the university. He's got his PhD at Maryland and is now a faculty member at University of Georgia. He's provided me with his modeling code for Bacillus cereus modeling. And so we're going to do Clostridium modeling, Bacillus cereus modeling, and then also compare to the FDA food code, and then look at that against these 99 cooling curves, with the idea of saying, okay, is this 2-point method valid? How long is a restaurant inspector usually in a restaurant? Let's imagine maybe that they came in at one time, and then let's pick another time point an hour later. Let's maybe randomly select those time points during those cooling curves using modeling and risk assessment techniques, and then determine whether those predictions actually give us any measure of safety or not. And so that's the next step in the cooling curve story. We also have it's not quite an editorial, it's a commentary that should be appearing in the NEHA journal, a National Environmental Health Association journal, that was co-written by Laura Brown-Green and Nicole Hedin and myself, just sort of talking about cooling in general and the 2-point method in particular.
Thank you. Yeah, that's great. It's very important, but sometimes we missed it, that cooling curve and all those things. Here's another question from Kumar. As we make the assumption in the model development, what is one of the surprise that evaded the model prediction, challenged the fundamental belief that we have about the microbial survival? And is there any interesting anecdotes?
Yeah, so there's one that I've published a bunch of papers, but I'm happy with all the ones I've published. I'm sad about the ones that I haven't found the time to publish yet. There is still one that deals with cooling again, and it has to do with the rate of, basically, so when you have a cooked food, you have spores in that cooked food. Those spores have been heat activated by the cooking process, and then those heat activated spores pass through a certain temperature range during which they're germinating, and then eventually they germinate, they turn into vegetative cells, and those vegetative cells can outgrow into cells that begin to multiply during the rest of the cooling cooling process. And so one thing that surprised me, and I still remember where I was not in Minnesota, unfortunately. I was on a highway driving home in New Jersey, outside the Brunswick Square Mall on Route 18, thinking about, I was paying attention, but I was also thinking about modeling. And it occurred to me that there was something different about the kinetics of spore germination and the kinetics of microbial growth. And it's too complicated to go into without some pictures and diagrams and waving my hands some. But basically, there's something fundamentally different about a spore that's germinating versus a vegetative cell that's outgrowing. And I still hope someday to publish that work, and to put those ideas, I put them on paper, I submitted the paper, it got rejected, and then I didn't do anything with it. So I need to dust those ideas off and get that submitted. And then I also want to say one more humorous Minnesota related anecdote. Again, eternal thanks to Frank Bushta for inviting me to be part of the IFT panel and all of the great things that came as a result of that. I still remember on a regular basis telling Frank about the work that we were doing with Clostridium perfringens or Bacillus cereus, and inevitably, I got to a point in the story where Frank would say, "Oh yeah, we did that." It turns out that Frank had actually done not quite the same thing, but he had done something very similar 20 or 30 years before. And so every time there were so many stories that ended with Frank saying, "Yeah, we did that," and then me having to go read the literature and then say, "Oh yeah, Frank actually did kind of do that. We should do something different and better beyond what Frank had done already." So again, just a huge thanks to Frank for inspiring me in all of the spore work and so much other work that we've done.
Don, This is Frank. Can you hear me?
Hey, Frank.
You're too kind. I have a question on whether you have looked at the potential changes in models as we go to clean labels and changes in ingredients that appear to be minor, including reduction of sugars, reduction of salts, etc., and what that's going to do to the models.
Yeah, it's a great question. I actually have the opportunity to have a really interesting data set. I can't talk too much about it because it's a consulting assignment, but let's just say it's from a trade association that has a lot of different formulated products with a lot of minor ingredients that, and so they've given me this wonderful data set. I'm in the process of analyzing it with some help from a buddy that's better at statistics than I am. And trying to figure out, like, so in this particular system, pH drives a lot of the inactivation, right? Acid moisture ratio drives the inactivation. And then in all of these products, there's so many ingredients, right? Like some have blue cheese, some have oil, some have vinegar, some have water. And so we've got this wonderful data set, probably 70 inactivation curves with all of these different components, okay? None of them the same. Some of them, like all of these formulations have water. Many of them have different kinds of acids. Some of them have different kinds of oil. And trying to make sense of all that and find out which of those ingredients actually drive inactivation rate. Because ultimately what this industry would like to be able to do is to go to the regulatory agency and say, hey, look, products that have this formulation with these ingredients, we can guarantee that they're going to be safe because they will have experienced the 5-log reduction in whatever your pathogen of interest is, Salmonella, E. coli, Listeria. And it turns out that it's complicated, right? It's complicated because of the variations in these formulations. Not everything has the same different ingredients. The trade, like to your question, the trade-off between green ingredients and maybe more traditional, less green ingredients, we just don't know. And so I think that the short answer is I think models can help us there. The longer, more complicated answer is we're going to need a lot better, more detailed models. And those just aren't out there. And the industry is collecting data, but they're not always thinking about modeling when they collect the data. And so the experiments are often not well designed from a statistical point of view. But yeah, so the short answer is I think models can help. The long answer is it's going to take us a while to get there. But I think we'll get there. Great question, thanks.
Great. Any more questions? We have plenty of time. Thank you, Dr. Schaffner, for leaving a lot of time. It's a great discussion.
I have a question. This is Ruth P. Tran. So to that point, Don, it's great to see you from afar.
Yeah.
You know, you're right. Industry does have a lot of data because it's part of doing hazard analysis and all that. You know, would you point to that MNIFT document as kind of the best guidance around how to best collect those data so that they are most useful?
Yeah, I would say that was the best advice that we could give at the time. That document is, it's hard to believe that the IFT document is more than 20 years old, and the MNIFT document is I guess we're getting on more than 10 years old, right? So it was the best guidance that we could give at the time. I think that some of our European colleagues are also getting involved in this, and they have guidance coming out. Has come out. I haven't been following it closely. I think looking at what those recommendations are, you know, and finding a way to harmonize that or figure out where the differences are is really important. And then again, I've alluded to Combase a little bit. I've mentioned Perfringens Predictor, which is part of Combase. I think that, so for those of you that don't know, Combase is a bunch of different things. It's some modeling tools, but it's also a data repository where people can put data from the literature. And I think that ultimately the real power here is if we much as we've seen with people sharing sequences of microorganisms and looking at how whole genome sequencing has revolutionized food safety, I think those of us that do maybe more traditional food microbiology, where we formulate products, we put pathogens in and we look and we see what happens, if we can take those data and put them into a common database like exists with Combase, that's going to help us begin to get at, like, maybe to Frank's question, what some of these more important factors might be, right? And that may guide us to your question, Ruth, in terms of how do we design challenge studies, where, and again, we've gotten really interested in this with respect to Listeria, and Listeria specifically in whole produce. We know that there's a lot of recalls in this country because of Listeria on whole produce. What we've discovered is, depending upon how you do the experiments, you may get apparent growth of Listeria in whole produce, but you may not. And it's really dependent on the way that you put that Listeria into the whole produce. We've got one paper out there and another paper that's going to be submitted soon that shows that the paper that's going to be submitted soon shows that the way that you inoculate it really matters. And so if I was going to sit on a NACMIF committee today and change anything, it would be around the idea of how do we put the organism, how do we culture the organisms before we put it in the food? We know that if we're looking for, if we want to bias the organisms towards growth in foods that support growth, we want actively growing cells. Well, if we're going to put those same cells into a dry food where they're going to survive, or maybe a whole produce where we don't know what they're going to do, maybe it's not the best thing to put them into growth media where they're actively growing. And then all of this sort of presupposes the question, well, what is the actual state of the organisms when they come into the food in the real world? Because you can do lots of things in the laboratory that are not relevant to the real world. So what's the state of Staph aureus when it gets into a filling on a baked good? Well, it's probably whatever state it was in when it was on the hands of the chef, right? That was making those baked goods. What's the state of Listeria when it gets on a hot dog in a food processing plant? Well, I don't know. Maybe it's been aerosol. Maybe it's contact with a food contact surface. And so setting up our challenge studies to reflect the physiological state of the organism when it gets onto the food in the real world, I think is a real challenge. And so, I mean, that NACMIF document is really good, but we could probably make it better. And we certainly should think about ways we could harmonize it with what our European colleagues are doing. And so, yeah, it's a really good place to start. Given that many people haven't even read it and they're doing challenge studies, a really good place to start would be to read it. But I think we could certainly go beyond it and do better as well.
Great question, thanks. And
I didn't get any Lutefisk references into the talk. Sorry. I did. I was going to talk about, speaking of cross contamination, how foods get contaminated. I was going to work. I had time. I was talking really fast. I could have worked in the work that we did on frozen hamburger patties, which was done for a large food service corporation that's not based in Minnesota, but the sanitizer that we used did come from Ecolab. And so I had time. I could have worked in an Ecolab reference, but I didn't. So, but there you go. Thanks.
Yeah, thank you. Any more questions for Dr. Schaffner?
This is Frank Bush again. Don, if on the dry challenges that you mentioned,
Mm-hmm.
you might want to go back way into the old literature, say late 60s, early 70s on dry milk, on non-fat dry milk. And maybe work that was done by Bibic Ray. That was a dry system, and the challenges of recovering salmonella out of that dry system. There might be some hints there of, this is ancient work with a lot, many of the new tools not available, but there were some aberrant things that occurred when you get into dry systems. Just
I've just made a note, and I will say, you know, it used to be that graduate students didn't think the literature existed before the 1960s. At least now, thanks to Google Scholar and other tools, at least it's all indexed and it's out there. And so if they don't find it now, it's their own fault. But yeah, I've made a note. So thank you. And if you've never, for those of you who are younger scientists on the call who've never read some of these older papers in the literature, they're an absolute delight to read. I think sometimes we've gotten a little bit too clinical in our presentations, but some of these old papers are really just a lot of fun to read. So dig into that older literature. You might learn something, and you'll certainly be informed by maybe a slightly different way of communicating science. So thanks for that, Frank.
Great. All right, I think so. We are almost at the time. So Dr. Schaffner, well, thank you once again for an excellent presentation, and it's so, you know, so much impact of your work actually. And so Dr. Schaffner has kindly agreed to, so we have recorded his presentation, so you can look it up anytime. We'll upload it or either on Minnesota IFT website, or we'll have a YouTube video or something like that, probably. Not sure how we're going to do it, but, and then also, I think you're going to share your copy of your presentation as a PDF copy as well.
Yep.
So we'll actually certainly upload that one on a Minnesota IFT website as well. But on once again, Dr. Schaffner, on behalf of everybody at Minnesota IFT, congratulations once again, and we are so glad that you can join us, and such a nice presentation.
No, thank you so much for the invitation. My pleasure. Thank you.
Thank you. All right, so I will then turn over back to Emily or Sonali. So I want to take it over, or Dr. Krishnan for a student recognition. So please stay with us, everybody. We have another event following up.